Maybe I can take it first. So I think there was an extreme situation during pandemics when, I think all the vaccine manufacturers were prioritized, which makes it a tough way to go for everyone else. But I think it was absolutely acceptable, and it was what we had to do in that range. Nevertheless, even after pandemics, we now see that supply chain is still a topic. It's now replaced from the prioritization during pandemics via the growing field in general, and I think in general global supply chain issues in the whole field. So there are lead times for, let's say, a sterile filter that might be 6 months or something like this, which is a difficult thing to address. If you don't know, it's difficult to address, but I think it's still always a timeline topic, and you just have to reflect it correctly in your timelines, in your Gantt charts, and especially if there are limiting factors inside, you can simply de-risk them via, first of all, a very good supply chain that has to, I think, be always with reliable partners, and I think we at Ethris have very good and very reliable partners. Open communication with them, and simply addressing these topics as early as possible in the projects to at the end meet all the criteria and do not delay the development in general. But it's topics that can be handled if you know what is limiting and what you need at which time.
Maybe if I can add to this, so during the pandemic, there was clearly bottlenecks on all levels. Yeah, it was really hard. But the upside is since then the whole manufacturing CDMO area has expanded tremendously. So new companies that doing, for example, lipid synthesis, even up to GMP, were popping up. The big players like Merck, Croda, and things like this. They were really investing into this field, and I think that's also generating, I think, in the future overcapacities, but also the knowledge in the field for such things really increased. So I think for some of the things, the situation is already quite nice, and you can select from several different vendors that can produce you high quality materials. For the consumables, it's clear there are still some hiccups due to the pandemic and due to some wars and other stuff that is putting pressure here. But usually as a company, at least if you are evolved a little bit, you would have a supply chain management in your company that really looks into vendors, making contracts with them and securing your supply.
Thanks, Patrick. Miles, let's take a look at those poll question results. Immunogenicity safety risk at about 26%. Producing high yields or high quality mRNA, 32.3%. So it looks like those are the two leading. And then temperature handling stability. We had a couple questions about that too, so I want to get your takes on where things are going in terms of cold storage and stability. But in the meantime, Christian, what do you make of these results? Anything jump out at you as a surprise?
Well, I would have expected such a rating. I think the immunogenicity topic we addressed already, but that's something to be addressed for all drugs you develop, I think, and especially, as you mentioned already, we'll take the temperature and the handling stability topic in a second, but I think that's a special topic to be addressed for mRNA drugs that can be optimized in future, definitely.
Mm-hmm. Patrick, anything jump out at you?
Yeah, also the things that I would have thought would pop up principally, yeah. And stability, clearly, I can comment on this then.
Yeah, let's do that. Let's talk about stability. What are the current challenges? I mean, obviously during the pandemic, the conversation was all about cold chain and how we're going to enable this global, cold chain supply to move vaccines all around the world. Where have we come since then? Have we made progress since then? And what does the future hold in terms of stability?
Yeah, I can take this. Usually, mRNA is quite a new technology, and if you develop a new technology, you usually try to maximize efficacy in the first place. So, and when the pandemic hit, we were just at a level where we had some phase 1 trials that were showing that messenger RNA is working in humans. And then some people embarked with no prior clinical trials, just using technology that was available. Others had some experience. But usually in drug development, you go through a phase 1, then into a phase 2. And during the transition from phase 1 to phase 3, you usually fix the other stuff, like doing stability trials, principally change maybe the matrix, or doing some other stuff to really get these parameters that is for market quite important in place. Usually this is a span of 3 to 5 years in development, and it was condensed to a few months. Mm-hmm. So it was clear that what was on the market was not going through these certain iterations that would usually fix or look into these things. So everyone in the field is currently looking now on fixing stability, that we can get to a suitable one-dose, single-dose pre-filled syringe in the best case that would be stable at 4 degrees for a prolonged time. But I think with the current vaccines, we are not there, and maybe we see then also in the future, maybe new vaccines that would fix these issues, then also directly designed to have the stability.
Yeah. Christian, what would you add to that?
I think for vaccines, it's important to come to more milder conditions, away from this minus 70, minus 80 requirement, more to, as Patrick said, fridge conditions, 4 degrees. But I think especially in the therapeutic field, it's even more important to have these conditions, because patient compliance, you want to have the patients having their dose at home, and most of them do not have a minus 80 in place. You want to distribute over around the world to be able to bring the drug to the patients. And I would even go there a step further and would state that ideal conditions would be room temperature conditions, because it would simplify everything a lot. Here we at Ethris again working on respiratory disease, working on therapeutic. In the therapeutic area, we addressed this quite early. We see that there are different factors that play a role here. Once, of course, the conditions and the buffer, the LNP is inside, which can have a large impact on that. But there are also aspects of the LNP itself that supports storage at milder conditions. We, for example, have a LNP formulation that's already very stable at minus 20, which simplifies the supply chain or the cold chain already significantly. In general, we are also working on a lyophilization process where we have, on an R&D basis, data that supports stability of currently half a year at room temperature without any loss of quality of the LNPs. But yes, stability studies are still ongoing, and the process hasn't yet been transferred to a GMP state and to CDMO. Furthermore, for respiratory disease, there are more options like dry powders that could lead to more stable formulations, but clearly in the therapeutic area, there's I would tend to say even higher need to develop these formulations, and therefore we are also tackling these aspects already in early development of new candidates.